Okay, welcome to 2.5. In this lesson, we're going to be putting everything that we learned about cells together. We're going to be referencing nucleuses and organelles, and we're going to learn about how the cell divides. So on the slide, I've shown you a cross-section of an onion root, I think it is, under a microscope. And when you take a look at cells under a microscope, you're going to see something like this. You're going to see a lot of cells tightly packed next to each other. What I want to draw your attention to is the fact that they don't all look the same. So some of them seem a little bit darker, and some of them look like this, which is a large jumble of things. And what this shows you is that each cell is at a different level of the cell cycle, right? And what we are going to be learning, hopefully by the end of this lesson, when you learn about the cell cycle, you should be able to look at a slide like this and tell me what stage of the cell cycle each of these cells is. And I'll show you a few examples of pictures of what it looks like for different levels of the cell cycle, what those cells look like when you look at it under a microscope. Okay, just to start off with, the life cycle of a cell can be separated into two sections. Number one, interphase. Number two, cell division. So interphase, which is the first stage of the cell cycle, is the longest part. So the cell spends most of its life in interphase and spends a very little time, a very less amount of time in cell division. This is a very, very short time of the cell cycle. So let's take a look at interphase into a lot more detail. So interphase is the growth stage of the cell cycle. So this is the part of the cell's life where it is growing. It carries out everything except for cell division. So every other thing that a cell does happens in interphase except for cell division. So these activities could include things like growing, cellular respiration, or any other function that particular cell does. So during interphase, a few specific things happen. The cell grows to nearly double its original size. So this is towards the end of interphase. As it's preparing for cell division, it grows to nearly double its original size. The number of organelles in the cytoplasm also double, right? This is in preparation for cell division so that whatever cells are separated, each have a set of those organelles. And lastly, chromatin in the nucleus. So if you remember from genetic material, that's what we call chromatin. During interphase, they're called chromatin. When they double and duplicate towards the end of interphase, we call them chromosomes. And chromosomes are what we are able to see when we look through a microscope and see genetic material, we see chromosomes. Interphase itself can be divided into three specific stages. So this is the longest stage as a whole, but within it, there are three sub-compartments, if you will. The first phase is the G1 phase. So during the G1 phase, the cell is growing. During the S phase, the DNA or the genetic material is synthesized. And during the G2 phase, this is where the cell is preparing for mitosis or cellular division. Okay, so interphase itself, so based off of that first diagram there, interphase itself can be separated into three specific parts, G1, S, G2. If you take a look at the slide that I attached to the PowerPoint, most of those cells were in interphase. How do I know that? Because if you take a look at those cells, you see the nucleus very clearly. If a cell is in interphase, the nucleus, usually those dark spots, are very, very clearly held together. That's not the case when we get to cell division, so I will talk about that when we do. But when you see a slide and you see that the nucleus is together, chances are it's going to be in interphase. Okay, let's get to the second stage of cell division. Second stage of the cell cycle, which is cell division. And cell division itself can be separated into two parts. One is mitosis and the other is cytokinesis. Both words that you haven't heard before, but we'll get into it. So mitosis, which is the first of those two stages, is the stage where everything inside of the nucleus duplicates and it's dividing. Cytokinesis is the stage where the cell itself, so including the cytoplasm, separates into two. So let me just clarify, I think I misspoke earlier. Mitosis is when the nucleus is divided, just the nucleus. So by the end of mitosis, you should have two nuclei. And cytokinesis is when you'll eventually end up as two different cells. So the cytoplasm and everything else around the nucleus is also separating. The result of which, so at the end of cytokinesis, you will have two identical cells. And mitosis, which is our first part of cell division, can be broken down into 4 stages. So prophase, metaphase, anaphase, telophase. We will go through those in a lot more detail as well. But just to recap where we're at so far, we started off with our cell cycle being separated into interphase and cell division. So our cells, the life of a cell, could be separated as interphase or cell division. Within interphase, you have G1, you've got the S phase, and you've got G2 phase. At the end of the G2 phase, we're officially into what we call cell division. Cell division can then be separated into mitosis and cytokinesis. So mitosis is our first part of cell division, and then that is then further divided into prophase, metaphase, anaphase, telophase, and then we get into cytokinesis. Okay, so just to recap, a cell's life starts off at the G1 phase, it goes into the S, G2, then it goes to prophase, metaphase, anaphase, telophase, and then cytokinesis. Okay, so you can see how this organization level happens. So the two main components of the cell cycle, and then those smaller compartments of what happens within each of those stages. Okay, so we're going to be looking at the different parts of mitosis in a lot more detail. So prophase, that's our first phase of mitosis. Within prophase, the nuclear membrane, that's what we call the outside of the nucleus, it begins to dissolve. So it just means to break down, and you won't see it any longer, which is why when you take a look at a cell and you see a nucleus, you know that it's not dividing. It's an interphase because that nucleus is still together. The first part of cell division is that nuclear membrane either completely dissolved or starting to dissolve. Secondly, you've got the duplicated DNA, right? We said that was chromosomes. It condenses and forms chromosomes that become visible under the microscope. So if you take a look at a slide and you can see the chromosomes, then you know that you're in some form of cell division. Each chromosome is made up of the two chromatids of DNA that are joined at one point, which is our centromeres. So if you take a look at those circles that are joining or acting as a joint of the two parts of the chromosome, that is what we call the centromere. A few other things that I want to draw attention to. So in the picture, you can see the nuclear membrane. I've labeled that there for you. If you could do that into your notes, that would be great as well. Secondly, you have these lines that are starting to attach to those chromosomes on either side. So all around the cell, you're seeing these lines form. These are what we call spindle fibers. And it's not mentioned, we haven't talked about it, but it is in our diagram because it comes into play later on. And these spindle fibers form in prophase. So they only start to appear in prophase. If you take a look at a slide under a microscope, this is not the best picture, but it is a picture where you can see the chromosomes. So in this particular case, you've got your cell. That's sort of the outline of that cell. And within that cell, the key part of this that's telling me that this is not in interphase is that I don't see the clear outline of the nucleus. Instead, I see these different lines inside of this particular cell that's telling me that those are more my chromosomes. If I zoom in, if this picture was just even a little bit clearer, I would see the chromosomes and the X shapes that are there, but they are mumbled and joined together in this sort of soup, if you will. But you are seeing you can see the chromosomes and you don't see a nucleus at all, which tells me that this is in prophase. Moving on to our second part of mitosis, which is metaphase. So in metaphase, what happens, it's pretty simple and it's also very clearly identifiable when you look in a microscope, is that the chromosomes line up in the middle of the cell. So exactly in the middle, or what we call the equator, the chromosomes tend to line up. And they line up such that one part of the X is facing one way and the other part of the X is facing the other way, right? So if you were to split the cell up, I'm just going to draw a line here. If you were to split the cell up into two this way, you'll have one of these parts of the X going one way and the other going the other way. And so all of these chromosomes are going to be lined up in the middle, and they're attached to these lines. If you can identify it, that is exactly the same lines that we saw earlier on in the first stage of mitosis, which is our spindle fibers. So these lines are able to hold the chromosomes in the middle in metaphase. Easy way for you to think about it, metaphase is M, middle is M. That's a key way for you to remember that in metaphase, all the chromosomes are lined up in the very, very middle. If you take a look at through a microscope, this is what you would see. So this is my cell. Oops. Just a rough outline of what the cell looks like. These lines here, or this dark shading that you find there, those are my spindle fibers, and they are holding on to the chromosomes, which I see here, and they're lined up exactly in the middle, right? So you've got a little bit here and a little bit there. That's just showing the parts of the chromosomes that are facing either way, but essentially they're all lined up in the middle. Moving on to our third part of mitosis, which is anaphase. In anaphase, the chromosomes are split and those chromatids move to opposite sides of the cell. So where we had our line here in metaphase, so all those chromosomes used to be lined up there. But now they're being pulled by our spindle fibers, which are our lines here. They're still connected to our chromosomes and they're pulled to either side of that cell. The separated chromatids are what we call daughter chromosomes, right? So the wording of that, so they were chromosomes as they were lined up in the middle of the cell. When they are split up to either side, they're called daughter chromosomes. This is what this looks like under a microscope. I'm not going to attempt to draw the cell outline again because I think it's pretty clear that you can see that that's where the cell is. These chromosomes are being pulled to either side of the cell. You have this dark shading there that clearly tells you that those are our spindle fibers and they're being pulled to either end of the cell. So that's anaphase. So prophase, you have our nuclear membrane dissolving and you can see those chromosomes. In metaphase, they're lined up in the middle. In anaphase, they're being pulled to either side of the cell. Okay, telophase, our last stage of mitosis. So after our chromosomes, daughter chromosomes, were pulled to either side of the cell, a new nucleus or a new nuclear membrane starts to form around each of those chromosomes, right? So you've got a new nucleus forming within each part of that cell. You've got two different nucleus. It appears as if you've got two nuclei. Even though that's not yet fully formed, you can still, that's what you would see if you were to take a look at a cell through a microscope. However, this cell has not separated into the two yet. You'll still see that the cytoplasm has not completely divided yet. That's because cytokinesis has not occurred. So this is what you might see in telophase. So these daughter chromosomes have collected together quite closely, and there is a pinching of the cell, so there will be a line forming there that might tell you that this is towards the end of mitosis. So the reason why you have these two nuclei within one cell might be because cytokinesis is about to happen. And in cytokinesis, the cytoplasm, which is this surrounding substance all around the cell, is splitting. And those organelles that we talked about way back in interphase, we said they were doubling. Those are now going to either end of the cell. So this will get, so for example, if there's a certain number of Golgi body in the cell to begin with, each one of these cells is going to have the same number of Golgi bodies. The cytoplasm splits so that you can have two cells, and the cytoplasm surrounds each one of those nuclei, right? So you're going to end up with two different cells here. This is a continuous stage, so all of this division, all the way from prophase all the way to cytokinesis, is a continuous process. So you might see when you take a look through a microscope that it might be a little bit hard for you to identify is that late telophase, early cytokinesis. So we can only guess based off of the clues that we are given. And often if I'm asking you to identify a slide and what stage it is, I will always ask you to justify. So tell me, oh, I see the spindle fibers, I see the chromosomes are separated, I know it's in this stage of the cell cycle. Okay, so in cytokinesis, there's a way more clear distinction between the two cells. And not only that, I can't see my individual chromosomes. Rather, I'm seeing a black dot or a black blob. That tells me that my chromosomes are no longer those thick chromosomes, but they're rather chromatin, right? So they're going back to the beginning of interphase. So in order for this cell to function, or any cell to function, there are certain checkpoints that need to take place to see whether or not the cell can actually go through with cell division. Right? So the timing and the rate of cell division in different parts of organisms, it's very, very crucial to the growth and maintenance of a particular organism. Right? So that means that we need cell division to happen at a faster rate if you have something like a wound, right? And an average cell, say in your skin, has to replenish itself after a certain number of days. So there is a time, there's a rate that is naturally present in our bodies when it comes to cell division. This needs to be maintained, and this is maintained through what we call checkpoints. So they're at certain parts of the cell cycle. So for example, in interphase, G1, S, G2, at the end of each phase, the cell is able to determine, based off of chemical signals that it gets, whether or not it has or it should follow through with cell division or not. Okay, so the three major checkpoints are found in our three parts of interphase, which is our G1, S, G2 phases. And by the end of that, the cell can determine whether or not it can go into mitosis or not. And how does the cell determine that? It's through chemical signals, that it receives, and then that can tell it to go on to mitosis and cytokinesis or not. That's pretty much it. That is a long and complicated lesson. If you need to go back and maybe listen to it one more time, these textbook sections are not these ones, I don't think. I've posted it on the outline page, so just check back to those and see what questions you have to do, and then submit it onto the classroom as well. Okay, bye!